Welcome to the lab tutorial on performing an EMSA. Before you begin, have your protein on ice with tubes labeled for all of your dilutions. You should also have your DNA stock protected from light, a tube for your DNA dilution, and tubes for your reactions. Start by pipetting the appropriate amount of load storage buffer into each of your protein dilution tubes. Note that I am leaving the lids open to indicate which tubes now contain buffer. Next, pipette the appropriate amount of protein stock into each tube, being sure to mix thoroughly each time. Note that for several dilutions, you will be pipetting from a diluted stock rather than the original stock tube. After adding the protein, I am now closing the lids of the tubes to mark that protein has been added. Next, dilute the DNA to the appropriate concentration by combining the working buffer and DNA stock solutions. Aliquot the diluted DNA into each of the reaction tubes. Note that I am leaving the lids up once I add DNA to indicate which tubes have solution added. Now we will add the protein dilutions into the reaction tubes containing the DNA. As protein is added, be sure to mix. Note that I am closing the lids and moving the tubes down a row to indicate when the tube has protein added. Place all tubes at 37 degrees Celsius to incubate for 10 minutes. Keep tubes covered to avoid light exposure for the fluorescein label. While the tubes are incubating, set up your TVE acrylamide gel. Acrylamide gel waste should go in the designated unwanted materials container. Be sure to remove the sticker from the bottom of the gel and remove the comb from the top. Place the gel in the gel rig. If you are running only one gel, be sure to use a buffer dam on the opposite side to hold in the buffer. Be sure to use TBE buffer, as this is a DNA gel. Pour buffer in the space between the two gels, then into the container up to the 2 gel mark. After the 10 minutes, remove the reactions from the incubator. Add load buffer to each, being sure to thoroughly mix with the pipette tip. Use the P10 to carefully load the purple dye and your reactions into the wells of the gel. Because the reactions are clear, you will need to carefully watch to see your reaction enter the well. Turn on the power supply. Place the lid on the gel rig and hook it up to the power supply. Set the gel to run at 150 volts for 30 minutes. Cover the gel rig in foil to avoid light exposure while the gel is running. After 30 minutes, stop the power supply and remove the foil and lid from the gel rig. Take the gel rig to the sink to pour out the TBE buffer and remove the gel. Carry the gel over to the ChemiDoc imager. You will need to break the plastic casing with a spatula to remove the gel for imaging. Place a small pool of water or buffer on the imager and use the spatula to slide the gel onto the imager. The gel should be placed off to the left side to avoid some scratches in the center that will interfere with our band quantification. On the computer, open the Image Lab software. Select Nucleic Acids, then Fluorescein from the drop-down menu. Select Position Gel and adjust as needed to be sure your entire gel will be captured. Select Run Protocol and the camera will capture the gel image with fluorescein emission. The bands may be quite faint, depending on how much the fluorescein has been exposed to light throughout this process. Use the black and white sun icon to invert the image so that there are black bands on a white background, and adjust the contrast as needed. Insert a USB drive and export the file for publication. You will need a TIFF file for the ImageJ program. Rename the file as appropriate. Don't forget to remove your gel and wipe down the imager. Thanks for watching!